A Chinese scientist is currently embroiled in controversy as they assert that they have created a 100% fatality rate mutant coronavirus strain in their laboratory. This research involves collaborative efforts from institutions such as Beijing University of Chemical Technology, Nanjing Medical University, and the People's Liberation Army General Hospital in China. They have created a virus mutation known as GXP2V and reported that when this variant was introduced to mice with genetically altered characteristics, all the mice died. Here are some key points. The original Qianzhanbach coronavirus does not cause any significant disease in humans. However, during the cultivation process, the research team claims to have created a mutated virus called GXP2V, which they believe can be lethal to humans. They conducted experiments using mice with modified genetic traits, expressing the ACE2 protein responsible for mediating coronavirus infection in humans. When they infected these modified mice with the mutant virus, all of them died within 8 days. The mice displayed symptoms such as weight loss and changes in eye color. Substantial amounts of virus RNA were detected in various organs of the mice, with the brain showing the highest concentration, suggesting that the virus targeted the brain by traveling through the respiratory system. This paper was authored collaboratively by Beijing University of Chemical Technology, Nanjing Medical University, and PLA General Hospital in Beijing. There have been concerns raised about the involvement of a military hospital in the research. However, the paper was published on bioRxiv, a preprint server that allows researchers in the field of biology to share their work without undergoing peer review. This raises questions about the reliability of the findings. The use of mice genetically modified to express the ACE2 protein, which is involved in coronavirus infection in humans, suggests that human transmission is possible. However, the presence of the ACE2 protein alone does not guarantee infection, and the severity of infection can vary among different species, even with the same coronavirus. Additionally, the accuracy of the experimental process and results remains unclear at this time. The scientific community has expressed concerns about the lack of clear records regarding biological safety levels and virus handling in the paper. If proper precautions are not taken, there is a risk of virus leakage. Detailed information on how experiments were conducted and whether virus-infected specimens were properly disposed of is missing from the paper. Expert opinions on this research are divided. While some see it as a warning about the potential dangers of virus mutations, others argue that since the research has not undergone peer review or validation, excessive alarm may not be warranted. It is noteworthy that the coronavirus pandemic originated in Wuhan, China, and research on the virus has previously been conducted in Wuhan laboratories.